Okay, class, in chapter 10 here, we're going to talk about post-transcriptional gene control. Okay, so there's a lot going on in this slide, so bear with me. I'm going to try to make as much sense of this slide for you as I possibly can. So, what we're going to look at is mRNA and how it is processed and how after transcription it can be manipulated to control the amount of protein made. So we're going to start here by just looking at polymerase 2, which is the polymerase that makes mRNA that are eventually going to be protein. And so, after transcription, we have this pre-mRNA, and there can be alternative splicing to combine different exons to make different variations of proteins or different proteins from a single gene. So here we are showing these introns are being spliced out, and then they are discarded. So then we have a spliced protein, has no introns, and we have a poly A tail that is added. And so now it's got the 5' cap, which is put on it during transcription, and the poly A tail. And from there, if the mRNA is processed correctly, it will then be exported from the nucleus to the cytoplasm. And then at this point, the ribosome will come and, with the help of some cap binding proteins, it will go in the initiation site of the mRNA and it will start transcription. Now, if the mRNA was not processed properly, back up here, let me change my color, back up here to number 3, then what happens is it gets degraded by these exosomes. So this could be if an intron wasn't spliced out all the way, or it was improperly spliced, or an exon is missing, or something of that nature, then it gets broken down. So once the mRNA is done being transcribed, it can be sent to what's called a P body. And this is a processing body, so P is just short for processing. And what happens here is there are enzymes that will come and it will take the cap off the mRNA, and it will take the poly A tail off. So we just have our ribonucleic acid with not processed or without the 5' or 3' alterations, and then it will degrade it. And so this is one way that we can control the amount of protein being created, is through this P body and degradation of mRNA. The longer that this mRNA is floating around in the cytoplasm without being degraded, the more proteins it can make. So if we want to make only a few proteins, then sending it to the P body to be destroyed after it makes only a couple will regulate how much protein. It will make a little bit of protein. If we let it linger a long time in the cytoplasm, then it will make more and more proteins. And so that's kind of a way, a switch, turn it up to 11 or turn it down to 0, so to speak, to kind of control how much protein is being made. So another way that we can regulate, or that mRNA is regulated to control the amount of proteins, is through the step 6 down here, the cytoplasmic poly A polymerase. Now, not always is a poly A tail added up here in the nucleus. Sometimes it is added later. And so there is this enzyme called cytoplasmic poly A polymerase. So all it is a polymerase that adds poly A tails to mRNA in the cytoplasm. And so this is a way that can be used to regulate how much of that gene is transcribed. Because if no poly A tail is ever added to this mRNA when it's in the cytoplasm, then this initiation of translation down here doesn't occur because there's this 5' binding protein, or 5' poly A binding protein, or excuse me, 3', that's the 3' end, 3' poly A binding protein that helps to get the mRNA in the proper conformation when transcription starts, or translation starts. And so if that poly A tail is missing, then we don't have that protein being translated. So this is another way that regulation can occur. And lastly, we have microRNAs. So microRNAs are 22 nucleotide RNAs, these very short RNAs, and they're complementary to the single-stranded mRNA that is on its way to being translated. And what happens is this will bind to a region of the mRNA before it gets to the ribosome, and that initiates this antiviral response mechanism called Dicer, and it uses drosha as this intermediate enzyme. We won't get too much into the weeds here, but what that does is it comes down and it breaks down those mRNA because it is a mechanism that recognizes double-stranded RNA as bad. And the reason for this is whether the chicken or the egg came first, but it's also used for viral recognition because a lot of viruses use double-stranded RNA as their means of replication. So this mechanism also breaks down RNA from viruses to protect cells. So whenever it sees mRNA that is double-stranded, it says bad, 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 breaks it apart. So we can make these mRNA or microRNA to target genes that we don't want expressed. And so in that instance, that's another way using this Dicer mechanism that we can stop the amount of protein being made or regulate it after transcription. So lastly here, we're mostly talking about the regulation of protein coding genes, but they also included, just to make the slide even more busy for some reason, polymerase 3 and polymerase 1. So polymerase 3 here, remember that makes tRNAs, so it transcribes in the nucleus. The pre-tRNA is processed and then exported. And remember, that pulls the amino acids as we're reading the mRNA and adds them to the growing amino acid chain. And then lastly, in the nucleolus, within the nucleus, we have polymerase 1, and this transcribes those rRNAs, and then they are exported to be integrated into the ribosomal subunits. Okay, let's all take a breath after that long slide there. Rub your ears, woosah, right? And calm down. All right, so moving on. Let's talk a little bit about how, get a little more into detail about how this mRNA is processed. So, for eukaryotes, cells convert this initial RNA polymerase 2 transcript into functional RNA by 3 modifications. There's that 5' capping, that 3' cleavage and polyadenylation, and RNA splicing. So we didn't talk about the cleavage previously of the 3'. We'll get into it here. So in step 1, there is this nascent unprocessed mRNA. And so here we are doing a beta globin gene, it says. It doesn't really say anything about that on the slide, but whatever. All right, so this RNA is capped on the 5' end with 7 methylguanylate, which is occurring right there. So that's step 1, transcription and that 5' capping. Now transcription continues past the poly A site. So here we have the poly A site, and that is the site that's going to let the enzymes that do the poly-A addition know where to add it, but the actual termination site of the transcription, there's multiple termination sites downstream of that poly-A site. And so that just kind of gives like a buffer at the end of the transcript to kind of protect that area. So once transcription is complete, we have these endonucleases come in, and they cleave off that buffer site that I just mentioned. So as you can see here, that extra long piece of RNA is now removed. And so all we have left is the poly-A site. Now, poly-A polymerase and ATP come in, and they read that poly-A site, and they do polyadenylation. So they add that poly-A tail. Now, in mammals, that poly-A tail is about 250 amino acids. In insects, it's about 150. So here I'll draw a little bug so you know that's insects. You can tell this quarantine is starting to affect everybody here. And then lastly, 100 base pairs or amino acids in yeast. I would draw a loaf of bread, but you will never be able to determine what that is. So 100 amino acids in yeast are added. And then in the case of very short transcripts, very short pieces of mRNA, then the introns are spliced out after the polyadenylation, the 5' capping, and the cleavage. Now, in very long transcripts, genes that have a lot of introns, that splicing occurs during transcription. So there's simultaneous transcription and splicing. So when you get to the polyadenylation and stuff, all the introns are already removed. So it depends on the length of the mRNA for this splicing when it occurs, either at the very beginning or at the very end. All right, here we have kind of a recap or a table of contents, as you will, of some of the RNAs that we're going to talk about in this chapter. We talked about mRNA and pre-mRNA, and we talked a little bit about microRNA and siRNA in a previous chapter, but we'll get into more of these. This is just kind of a definitions page for this chapter. Okay, so now let's talk about the next one down on our previous list, these HN RNAs. And HN RNAs are heterogeneous nuclear RNAs. And these are the proteins from these mRNAs associate with pre-mRNAs to prevent formation of base paired secondary structures. Remember, this is single-stranded, so those bonds are looking to be fulfilled, and so these come in to make sure they don't form improper bonds between the base pairing, and this makes the pre-mRNAs accessible for interaction with other RNA molecules and proteins. So it's kind of preventing them binding from stuff that's not supposed to be binding to them. So an example of these are proteins, or HNRNA protein A1 and HNRNA protein C. And these bind preferentially to the pyrimidine-rich sequences at the 3' end of introns. And some of these RNAs remain localized in the nucleus, while others can cycle in and out of the nucleus and help in the export of these RNAs from the nucleus to the cytoplasm. And so looking at the structure here of these HNRNAs, it's a mouthful for me for some reason, there is this motif that is found on them. And this is called an RRM, which is a RNA recognition motif. And these are about 80 amino acids, or 80 residues, we say. And it's the most common RNA binding domain in these HNRNP proteins. and other RNA binding proteins. And they contain two highly conserved sequences, RNP1, let me get my marker, RNP1 and RNP2, and these sequences are conserved all the way from yeast. That's probably a bad color, you can't really see it there. There we go, blue. Conserved from yeast to human, so it's highly conserved throughout eukaryotes. So it evolved very early in eukaryotic evolution. So the structure of this RRM domain is two alpha helices that are in the green, right here, one there and one kind of behind there, and four beta strands, which are red, including those RNP1 and RNP2. The two RRM domains in Drosophila sex lethal, or our second example here, B. As you can see there, we have our ribbon structure and then our 3D structure, and they're kind of mapped, RRM1 and RRM2. Together, these bind a continuous 9-base sequence in pre-mRNA, which is yellow here. So those domains, as you can see, are what are responsible for binding this pre-mRNA, so non-processed RNA. And that is to, remember, prevent this 3D structure from occurring that's not supposed to happen. Then lastly, as you can see in part C here, this polypyrimidine tract binding protein, or PTB, has those domains, the RRM3 and RRM4. There's still these RRM domains, but they are facing more outwards. And so you see that the binding of the mRNA occurs kind of on the outside of the protein. And so the conformation is different depending on the HN RNP, or ribonuclear protein. So I've been using RNA and RNP kind of interchangeably here, but HNRNP stands for the ribonuclear protein. So depending on which protein it is from this family, the RRMs can be in different orientations, either inward or outward, and they bind different mRNA. So how does this processing machinery know where to splice introns and exons, and what is an intron and what is an exon? There are these two trans-esterification reactions that are present in these short conserved sequences that lead to the splicing of these introns. And so the intron splice site has this very distinct conserved sequences, and it's called the AGGU rule. And so there, as you can see here at the splice site, we have an AG followed by a GU in the mRNA. And then if we were to look down at the 3' splice site, we have that again, AG, G, and it doesn't have the U here, but the U would be right there. Within that intron, 20 to 50 bases from the end of the intron, there is this conserved A residue. And this conserved residue is the branch point for this. So it'll end up forming this loop that we'll get to in the next slide here. And so the polypyrimidine tract near the 5' end here, so there's a lot of pyrimidines that are coded for in this region, is found in most introns, and the central region of the RNA is usually 40 to 50 kilobases, so it can be, or 40 bases to 50 kilobases, so it can be very short to very, very long, and only 30 to 40 nucleotides at each end of an intron are necessary for splicing to occur. So this double line here can be super short, or it can be super long, and it doesn't really affect the splicing because all that's required are these two regions at the very beginning and the very end, and everything in the middle is just there. Okay, let's talk about how this process of RNA splicing occurs. And so there are two transesterification reactions that result in the splicing of exons in this pre-mRNA. And so the first one occurs between that conserved A and the first phosphate group of the intron. And so what occurs then, as you can see here, is that there is a bond between that conserved A and the phosphate group, and it is no longer attached to that first exon. Since this oxygen is freed up now, it can go and bind with the first phosphate group of the second exon. And when it does that, it releases that intron that forms what we call a lariat or a cowboy's lariat. So you can see it kind of looks like a lasso. And then we have two exons that are spliced together and the intron's removed. Now this doesn't happen alone. There are RNAs that help in this process that we'll get into in the next slide here. Okay, so there are 5 uracil-rich small nuclear RNAs, sRNAs, that are associated with this process of intron splicing. And we denote them as U1, U2, U4, U5, and U6. U3, who knows where that went? It's probably a non-functional one, but is 1, 2, 4, 5, 6. And they're about 100 to 200 nucleotides long. And what they do is they bind to some of these recognition sites within the mRNA. So U1 up here, it base pairs across that 5' splice site, the intron-exon junction there. And U2, it base pairs with the sequence that surrounds that conserved A, that branch point sequence that's used to remove that first, or the front side of that cowboy's lariat, or that intron. Okay, so all the proteins, the RNPs, so ribonuclear proteins, which is a protein RNA complex, that both of those groups that are involved in splicing of introns is collectively known as a spliceosome. So we're going to talk about the mechanism of how a spliceosome works. And so it starts with this initial complex where we have U1, which is that RNA, that snRNA, associated with a protein, so now we call it an snRNP, and that binds over the intron-exon boundary on the 5' end. In addition, we have this protein SF1, which is splicing factor 1, which binds to that conserved A branch point. And then beyond that, we have U2AF, which is the U2 SNRP, SNRNP. They're referred to as SNRPs to make it easier on people like me that like to have word scrambles. So the U2 SNRP association factor is U2AF. So it's a protein that associates with that U2 SNRP. And so this U2AF associates with that polypyrimidine tract. So remember, there's that conserved region of pyrimidines that can vary in number. So here we have that Y to the nth, n can be any number. So that's that polypyrimidine tract. And the AG of the AGGU rule down on the 3' exon. Okay, so from there, that's our pre-initiation complex. We have step 1. In step 1, the U2 SNRP comes in and it associates with that polypyrimidine tract and the poly A site, or the conserved A site, and it displaces SF1. So SF1, that's out of here. and now U2 comes in, and it covers that that polypyrimidine tract and that conserved A branch point. Now in step 2, U4, U5, and U6, which we call a trimeric SNRP complex, that comes in and it binds to the spliceosome. So you can see kind of in between the two we have 4, 5, and 6, and they kind of fill the gap that was present right up here. So now in step 3, the SN-RNA base pairing interaction starts to rearrange. And so there is this catalytically active conformation that occurs. And so when U1 and U4 leave, that frees up U6, which was previously in the middle, U2, and U5 to come together and kind of bend this RNA. And as you can see, by this occurring, we have brought closer that branch point, which is, let me get this green here, right over here. We brought that closer to that AGGU on the 5' end. So now in step 4, we have our first trans-esterification reaction, and U6 and U2, they catalyze the formation of this interaction. So these two here catalyze the formation of bringing that bond together. And then the second trans-esterification reaction then occurs at stage 5 here. Oops, sorry, that was a long arrow. And this joins the two exons by this standard 3,5 phosphodiester bond, if you're really into the chemistry. And this releases the intron as a lariat structure. So as you can see right below here, we have this cowboy's lariat. It looks like a lasso. And that also, when that bond occurs, it releases the remaining SNRPs. So U2, U5, U6, they're out of there because they don't have any more reason to be there. And what happens then, so our splicing's complete, but we still have some things remaining, right? So we have this cowboy's lariat that's just kind of floating around in the nucleus at this point. And so what happens to that? So we have this cowboy lariat here, and a debranching enzyme comes in and it breaks this apart. So it linearizes that RNA again. And then once that RNA is linearized, it can be degraded and broken down back into the original nucleic acids and then recycled for further RNA production. So we've mentioned previously the carboxyl tail domain. This is a chain of amino acids, which is about 130 nanometers long. It's quite long, and it has multiple repeats of a 7 residue peptide sequence. So it's 7, then the same 7, then the same 7 over and over and over again. And this is where a lot of those U2 and the splicing factors, the spliceosome, is also the capping enzymes, all these things use this tail domain to come in kind of aggregate near the polymerase. Because remember, this polymerase is moving. And if you're an enzyme that needs to modify that mRNA, then you need to be moving as well. So the best way to do that is to kind of just latch onto it, right? So you can come in, if you're an enzyme, say this is U1 and U2. So then when this mRNA is coming out, they're right in the right spot to do their modifications that they need to do. And so that's kind of the point of the carboxyl tail domain. It also allows to bind the 5' cap, which would be at the top here, etc. So we talked about these introns, and in eukaryotes, the intron is on average about 3,500 base pairs long in humans. And the longest intron is over 500 kilobases, which is 500,000 bases long. That is a long, long way in a lot of sequence. Now, remember, our rule to define where splicing occurs is this AGGU rule. So those 4 nucleotides in a row, AGGU, is where the spliceosome recognizes that there should be a splice. Now, thinking about that 500,000 base pair intron, the odds are pretty dang good that in that sequence, there's going to be a random combination of AGGU in a row because we only have 4 options of nucleotides. So just by chance, we should have multiple AGGUs within that 500,000 base pair sequence. So how come we don't have things screwed up in these large introns where we have splicing in incorrect positions? So there is a mechanism for genes that have very big introns that prevent splicing in incorrect locations. And this is the work of these proteins that are called SR proteins. So you can see them right here. And SR proteins are proteins that their interaction domains are rich with arginine, which the abbreviation, the single letter abbreviation is R, and with serine, which is S, hence the name SR proteins. So these SR proteins interact with these exonic enhancer sequences, those ESE. So exonic meaning from an exon, and enhancer sequences. And so once they interact with these exonic enhancer sequences, they are able to bind and bring in U1 and U2 to the proper splice site as well as to the branch point. So as you can see here, these SR proteins bind with U1 with this little arrow here. They also bind with these kind of intermediate proteins as well as U2AF and U2. So what they're doing is they're saying this is a real exon. So when you find AGGU and we're nearby, that's the key to being able to bind in the proper location. The odds of AGGUs as well as multiple of these exonic enhancer sequences being present in random introns is very, very rare. So while having 4 base pairs like AGGU in a row is possible and likely in a very, very large intron, having those coupled with these SR binding locations is very rare. So that's how we prevent errant splicing in introns. So as with anything in biology, there's always exceptions to the rule. And so there is a class of introns that are self-splicing, that don't need the spliceosome to be removed. And so there are 2 types of these self-splicing introns. There are group 1 introns that are in actual nuclear genes, so in the genome of protozoans. And there are group 2 introns that are in protein-coding genes in the mitochondria and chloroplasts of plants and fungi. And what they do is they fold into a conserved complex secondary structure that contains a bunch of these stems and loops. Let me get why is my arrow not working? There we go. Sorry, I've drawn a whole bunch of lines here. So these stems and loops bring the 5' and the 3' ends together here and allow for this kind of splicing action to occur. So you can see the bond would be here, and this big long piece here, the intron, would be removed. Now, if you were to line up the RNA from those RNPs, you can see that the combination of all of these U1, U2s over here, and then 4 5, and 6. If you were to line these up, they look pretty similar to the group 2 introns, the self-splicing introns. And so the theory is that the spliceosome, the RNA in those RNPs, that evolved from the self-splicing introns. And so way back in the day, in ancient lineages, these introns splice themselves, and then since then they've been more efficient, I suppose, or some sort of selective pressure to say that they should evolve into these RNPs, and splicing should occur that way. So there is, like I said, there's always an exception to the rule. So some introns splice themselves out, while the majority of introns use the spliceosome. So let's get into more detail about the cleavage of this downstream mRNA that is after the poly A site. So almost all eukaryotic mRNAs have a 3' poly A tail. Histone mRNAs are the exception, and we're not going to get into that right now. So the 3' cleavage and polyadenylation of these pre-mRNAs are really highly coupled. So they are always associated together. And let's look at the mechanism for this. So this CPSF, which is cleavage and polyadenylation specificity factor, bind to this poly A signal, this AAU-AAA, which is upstream of the poly A site. Then this other protein comes in, or protein complex, CSTF, will come in and it interacts with a downstream GU. So as you can see here, it's the blue, and it will bind to CPSF. And so what that does is it brings those two together, and it causes the formation of this loop in the RNA. Then CFI comes in with CF2, or CF1, sorry, with CF2 comes in, and it stabilizes this bend. So it's just there to make sure nothing happens to that bend there. At this next point here, PAP, which is poly A polymerase, that comes in and fits kind of into the middle of these three complexes. And this stimulates the cleavage at that poly A cleavage site, and which is right by the poly A site there. So as you can see here, we now have a phosphate and a hydroxyl group. And once that occurs, the downstream mRNA, downstream of the poly A site, and these proteins that were responsible, or protein complexes that were responsible for the cleavage and bending of the mRNA, are all released. So CSTF, CF1, and CF2, they all leave, and all we have, and in addition to that downstream RNA, which is degraded at that point, so it's recycled for future mRNA. Then we have a 5' cap, and we have this OH overhang, and at first we have this slow polyadenylation step. So PAP, which is poly A polymerase, comes in and slowly adds about 12 A residues from ATP at a slow rate to that 3' hydroxyl group. And then after 12 or so of these A's are added for the poly A tail, another enzyme, which is PA-BPN1, which is poly A binding protein, and then N1 stands for, it's localized in the nucleus, so it's nuclear poly A binding protein. That binds to that 12 residue A tail, so the short A tail, and it very quickly accelerates the rate of addition by PAP. So it's there, it's kind of like an enhancer of PAP's activity, and then it will add up to 200 to 250 A residues. So we have this really long poly A tail after this occurs. So here we have another figure that's a little bit convoluted, so I want you to kind of bear with me here. And so we've talked about alternative splicing throughout this lecture and throughout this course. Alternative splicing in some organisms is an aspect of sex determination. So in Drosophila, which is the fruit fly, we have alternative splicing that leads to male and female development. And so in this figure here, the boxes are exons and the lines are introns, and that's all that's shown. There's no upstream UTRs, upstream or downstream. And in this case, anytime there is a line within an exon, for example, right there, that is a stop codon. And so in our example, we have our first protein here, which is sex lethal, and there is an intronic splicing silencer. And so this is present only in female embryos. There's no functional SXL or sex lethal protein expressed in males. So what happens is in males, we have the inclusion of, actually I should draw my arrow here, of this third exon that has that premature stop codon. So when this becomes a protein, what's going to happen is we're going to have a non-functional protein, right? Because it's missing part of it because there is that premature stop codon. In females, we do not include exon number 3, and so we only have 2 and 4, so we're going to end up with a fully functional protein of some sort. So then, when sex lethal is fully expressed in females, it is going to act as a transcription factor for the TRA gene. And so in females, we're going to block the use of exon number 2 in TRA. And so females will then have TRA only with exons 1 and 3, which leads to a fully functional TRA protein. Males, because they don't have sex lethal, are going to have all 3 of those exons. And because of this, in exon 2, they have another premature stop codon. And so again, just like with the sex lethal gene, they're going to have a protein that's missing part of it, and it's not going to be functional. Now, we finally go down to DSX, and DSX is a gene that leads to the expression of male and female proteins. So this has all been a cascade to get to DSX. And so in DSX, with the combination of RBP1 and TRA2, this female TRA protein that was expressed only in females is able to come in and bind and promote expression of the 4th exon of the DSX gene. And there can be any number of these repeats of this 4th the poly A tail on this 4th exon here. We had talked about that in a previous slide. So this TRA, RBP1, TRA2 combine to lead to the inclusion only of exons 1, 2, 3, and 4, which leads to the DSX protein female version. And in males, we have exon 3, and since there is no interaction of TRA, RBP1, and TRA2, exon 4 does not get included, and we have 3 and 5. So this leads to the male version of the DSX protein, which will then go on to express genes leading to male development. So alternative splicing isn't only a way to make different versions of very similar proteins, and can lead to this big cascade that can lead to differentiation in sexual organs and sexual development as well. Okay, so looking at how TRA, TRA2, and RBP1 allow this differential splicing to occur. TRA, TRA2, and RBP1 interacts with the double sex pre-mRNA, and RBP1 and TRA2 are these SR proteins that we talked about previously, and they require TRA to cooperatively interact with this exon. So they need TRA up here to be able to kind of form this complex that will interact with exon 4. So then in females, TRA, RBP1, and TRA2, this complex at the top here, that will activate exon 3-4 splicing by interacting with these 6 exonic splicing enhancers. So within this exon, there are these 6 regions that are conserved to allow that complex to come and bind. And in males, there's no TRA. So RBP1 and TRA2, they can't form this complex because TRA at the top here, that is the glue that holds together that complex. And so if RBP1 and TRA2 are floating around and there's no TRA because they're in a male, then that means that they're just kind of separate and they won't come together. And so that is one of the kind of stopgaps to make male development and not female development. And so when that happens, since there's no binding to these enhancers in males, exon 4 isn't spliced into the protein, and instead we have 3 and 5 that are spliced. As you can imagine, with any complex biological process, there are abnormalities that occur and will become a genetic disease. And so here is a link or a table of the genetic diseases that are associated with aberrant RNA splicing. So you see some that might be familiar, Duchenne muscular dystrophy. We also have Rett syndrome. Some of these I'm not super familiar with, as I am not a medical professional. But some of these you may recognize. So this is just for your information that aberrations in this process can lead to diseases. So we've talked about how alternative splicing can lead to all these different isoforms of a protein or different versions of a protein being made. And so this is one of the most extreme examples that we know to date. This is the Drosophila Dscam gene. And the number of exons and alternatively spliced exons to this is pretty astounding. And so there are up to 38,000 plus possible isoforms that are made from a single gene. And so as you can see, the different combinations of all these different exons, we have different that we classify into groups. We have this group of 12, this group of 48, and this group of 33 that can be combined in different combinations to make these different isoforms. In this example, the Dscam gene, it helps to specify tens of millions of different synaptic connections in the brain of Drosophila. And so as you can imagine, the brain's very complex and you need different regions and different connections, need different proteins, and thus this gene has so many different isoforms to be able to fulfill that need of neurological development. So let's look a little more in depth into how mRNAs are exported from the nucleus. So we're going to call this an mRNP, which just like we talked about the nuclear RNPs earlier, an mRNP is a messenger RNA that is in complex with all these proteins. So these mRNPs are transported through these nuclear pore complexes. And so this is a pore or a channel in the nuclear membrane that goes to the cytoplasm. And so there are these heterodimers, or large NXF1/NXT1 complexes. Heterodimer meaning there's two different subunits, so one is NXF1 and one is NXT1. NXF1 binds RNA and proteins in this mRNP complex, including this REF protein that is associated with the mRNA, and this stands for RNA export factor. And so as you can see, there's a bunch of other proteins also involved here in coding this mRNA. And this mRNP interacts with unstructured protein domains that will fill this nuclear pore complex. You can kind of see all this stuff within the nuclear pore complex there. So during this mRNP export, some of the mRNP proteins dissociate from this complex before going through. So they're just there to kind of bring the whole complex to the nuclear pore, but they never leave the nucleus. And some of them, such as NXF1, NXT1, and REF, dissociate once they've completed the job and move that RNA to the cytoplasm. Once this mRNP is in the cytoplasm, there's a couple replacement proteins that come in. So originally in the nucleus, CBC, which is cap binding complex, binds to that 5' cap. Once it gets to the cytoplasm and is successfully exported, this ELF4E replaces it. And similarly, in the nucleus, there are these proteins that bind to the poly A tail, which is PABPN1. But once in the cytoplasm, that is released, as you can see here, and PABC1 replaces it. So there's kind of a swap that are cytoplasm or nucleus specific. Now, all these proteins here that are released in the cytoplasm are then shuttled back into the nucleus through a nuclear pore complex and are then recycled and able to be used to export more RNA. Okay, let's look at how these proteins move to and fro across the nuclear membrane here. So we're going to look at MPL3, which is an adapter protein that is used to move these mRNPs across the membrane. And so as you can see, it's phosphorylated here to start, and MPL3 will then bind to this RNA as it emerges from RNA polymerase II. And then from there, after the polyadenylation of the tail here, GLC7 comes in and it removes that phosphate group. And so once that's removed, it allows NXF1 and NXT1 to bind to NPL3. Once that occurs, it can now be exported across this nuclear pore channel here, and it is in the cytoplasm. Once in the cytoplasm, this cytoplasmic protein SCY1 is able to re-add that phosphate group across or onto NPL3 here. And by doing so, that releases both NXF1, NXT1, and NPL3. Now, the mRNA goes down and is translated in these released NXF1, NXT1, and MPL3 with the help of a catalyst or an enzyme called importin, bring that back into the nucleus. And as you can see, we're in our starting state where MPL3 is phosphorylated again, and NXF1, NXT1 they don't really have conformational changes, but they're ready to go, and we can do this over and over again. And so this phosphorylation is kind of the switch that says stay in the nucleus or go to the cytoplasm. So let's talk a little here about degradation of mRNA after it has been translated into a protein. So there are mechanisms that control the stability and localization of mRNAs and the translation of them into proteins. And there are several mechanisms that lead to the abundance or regulation of these mRNAs. And in most higher eukaryotes, the mRNAs have half-lives of many hours. However, depending on the mRNA, there are some that are much less stable. So there's kind of a wide variation in the shelf life of an mRNA. And so there are three different pathways here that lead to mRNA degradation. There is the deadenylation dependent mRNA degradation, which is the most common pathway for degradation of mRNAs. And so within this, a deadenylase complex comes in and it shortens the poly A tail to less than 20 residues, and it destabilizes the PAB PC1 binding of this on the 5' end. And once that happens, the mRNA can be decapped by this complex of DCP2 and DCP1. As you can see here, this cap has been removed from the mRNA, and then XRN1, which is an exonuclease, comes in and it starts to chew away and break apart this mRNA. Now, the other way that this can occur in the deadenylation pathway is that no exonuclease comes in, but instead we have a 3 to 5 exonuclease that is present in the exomes in the cytoplasm that will chew it from the poly A tail side and will then break it down into its nucleotides. Now we also have a deadenylation independent mRNA decay, and in this case there is decapping that occurs before the deadenylation, and so the DCP2, DCP1 will decap before there's ever removal of that poly A tail, and then the XRN1 will come in and it will degrade in a 5 to 3' exonuclease direction while this poly A tail is intact. And then lastly, we have an endonuclease-mediated RNA decay. And this is where exonucleases come in and essentially chop up the mRNA into smaller pieces where both XRN1 and the exosome-mediated decay can both take place. So another way that protein synthesis can be regulated is through the mTORC1 pathway, and this has to do with translation initiation factors that are regulated as well as ribosomal proteins through modifications such as phosphorylation. And so mTOR is a mammalian protein kinase, and it responds to multiple signals from cell surface signaling proteins. So it kind of senses the environment through these these cell surface proteins as well as nutrient concentration. And so when we have an active TOR, so we're going to focus on the left-hand side here first, there's an Reb GTP complex that is activated. And this increases the rate of protein synthesis. So it phosphorylates this inactive 4E-BP complex, which releases the 4E component. And the 4E then stimulates the initiation of translation of genes. And it also activates S6 kinase. And S6 kinase phosphorylates ribosomal proteins that will stimulate the translation of mRNA then. So it kind of activates those ribosomal proteins. It also activates transcription factors for the different RNA polymerases here. So here we have polymerase 3, but also it will stimulate polymerases 1 and 2, which increases the synthesis an assembly of ribosomes, tRNA, translation factors, and all these other things that are required to kind of ramp up the machinery for production of proteins. And this active mTORC1 will also stop autophagy, so we're not going to be breaking down proteins in any way. We're going to be accumulating proteins. And if you were to get rid of mTORC1, all of these processes are then inhibited. So now in an inactive state, there is generally low cellular energy. This could be lack of nutrients, and so we're not going to ramp up production. And so we start with AMP kinase up at the top, which senses this low energy. And this activates this TSC1 and TSC2 complex, which then goes and inactivates mTORC1. We then have, it can also be activated instead of by low energy by stress response, hypoxia. Hydrogen peroxide accumulation and things of that nature lead to this stress response pathway being activated, which then goes and activates TSC1 and TSC2. We also have low nutrient concentrations that will lead to inactivation of mTORC1. So if there's not enough stuff to actually make proteins, the nutrients required for this process, it'll be inactivated. In addition, there is phosphorylation of this GTP-Reb to GDP-Reb, which will inactivate mTORC1 as well. And so these are ways that you can see there's kind of a sensing pathway that either blocks protein synthesis or enhances protein synthesis based on kind of the environment the cell is in. So lastly, for this chapter, we're going to look at the iron-dependent regulation of mRNA translation. And so there's kind of the sequence-specific mRNA binding that controls translation of certain mRNAs important for iron uptake. And so iron response element binding protein is kind of the switch here, this IREBP. And this controls the intercellular ion concentration by regulating the amount of ferritin and transferrin receptor mRNA protein. And so why is this important? Because iron concentrations are important since iron's a cofactor for a lot of enzymes, so it's required for a lot of enzymes to function. And if we have too much iron, this can generate free radicals that damage the cell or macromolecules within the cell. And so ferritin, the role of ferritin is that it is a intracellular molecule that binds up excess iron and kind of stores it. And transferrin is a membrane protein that brings iron into the cell if there's low concentrations. So ferritin reduces the amount of iron in the cell, and the transferrin increases the amount of iron within the cell. So in concentrations of high iron, we get the expression of ferritin mRNA and the creation of this ferritin protein, which will bind up iron and store it. Once that concentration is sufficiently low because of the activity of ferritin, then this IREBP, the iron binding protein, will become activated because it is sensing that there's a low concentration of iron, and it will stop the creation of ferritin because we don't need to store any more iron. It's sufficiently low, we have no more need to store it. Now, the transferrin kind of works in the opposite way. So in the high iron concentrations, we are not binding our IREBP to these IREs. Instead, we're allowing transferrin to this transferrin receptor mRNA to be expressed, When the concentration of iron is high, these iron response elements are unbound by IREBP, and this leads to increased degradation of those TFR proteins, or RNA, and thus less protein. So because we have high iron, we don't need to import more, so we're going to sense the concentration and then break down excess import proteins. When the concentration is low, then this activates these IREBPs, which bind to the iron response elements in this gene, and that prevents the degradation of those channel proteins, and so it will bring in more iron because we need more iron. So that leads to increased expression of genes. And so this is another way that protein expression is monitored and modified through iron sensing. So that's all we're going to do for this chapter. We've already gone over an hour and we've only gone through 28 slides, so we've cut out some things here, but we hit the main important elements, and I will see you in chapter 11.